Hello, everyone. Welcome to week number 5 in Bio 120. We're more than halfway through the course, so that is very good news, but we have some more to go. So let's figure out what's going on. Let's get to the next slide. So last week you learned about metabolism in the form of cellular respiration, which is a way that organisms break down food in order to produce energy molecules called ATP, adenosine triphosphate. And that energy in ATP is transferred to enzymes that do all the work in cells. And you learned about aerobic and anaerobic versions of cellular respiration. You also took a short little look at photosynthesis, which is harnessing sunlight energy to put together carbon dioxide molecules in water to make glucose. Glucose is the most universal food source on the planet that ever has been. It's a monosaccharide, which means a simple sugar, C6H12O6, and any organism basically can use glucose. All right, so this week is all about DNA. So that is one of my favorite subjects. Oh no, I thought I said that about cells. I think I did. Anyway, DNA. So read chapters 10, 14, and 15. Now, when I say read, I mean skim through them, okay? Skim through them quickly, see what's there, see where everything is so that you can go back and find the information quickly. Chapter 10 is all about cell division and how cells divide, and I'm going to talk about the cell cycle in just a second with that little diagram. Chapter 14 is about the discovery of the DNA molecule. DNA. The DNA molecule, everybody knows what it looks like. And also how it replicates, because that is a very important part of why it looks that way. And chapter 15 is about how proteins are made from all the DNA that's found in cells. And that's really the main job of DNA, to make those proteins. Most of the proteins are enzymes. Enzymes run all the biochemistry in cells. Okay, so that determines what kind of organism you are. Read the Module 5 overview and watch the videos. They will help you answer your questions this week. Your discussion board postings are all due by Sunday evening this week instead of having that Thursday deadline, because Thursday, of course, is Thanksgiving this week. So everything this week is due by Sunday evening before midnight. And your discussion board postings go like this. So if you're talking to a friend and you're discussing cancer, your friend might ask, well, is cancer a problem with cell division, including mitosis, which I'm going to get to in the cell cycle, or is it a genetic disease? Is it a disease of genes, or is it both? Now, last week I told you, if you ever see a question like this and somebody says or both, you know the answer is or both. Otherwise, they wouldn't even ask that. So anyway, it has to be both. And how might you change some of your lifestyle in order to lower your cancer risk? You could never say that you have zero cancer risk, because that just is not true. You are exposed to environmental carcinogens. You have genetic predispositions to certain cancers. So you can't get rid of every risk, but you can lower your risk. And then in your response post, you're going to talk about environmental carcinogens and how we can remove those from the environment. Okay, so let me get back to the cell cycle, because, part of that question in the discussion board posting is cancer a problem with mitosis? So I want you to look at the cell cycle over there on the right. It's a little circle that represents the life cycle of a cell. Okay, and we start right up there at that 12 o'clock position. That's the moment when this brand new cell is produced, okay? Then the cell moves into what's called G1, the G1 phase. G1 you could consider as a growth phase. Now, G didn't used to stand for growth. It used to stand for gap, because we had no idea what was going on. But as we've studied cell cycles more, we understand what's happening. So G1 is general growth. So the cell grows in size, the number of organelles reproduces, the number of enzymes ramps up really big, And so it has to reach a certain size. And then it moves into the S phase of the cell cycle. The S phase is the synthesis phase. And synthesis is the DNA is copied, is replicated. So what's synthesized? The DNA is. You have to make a copy of the DNA in order for the cell to divide. Then you have the G2 phase. That's a second growth phase, and this is where the cell division machinery all gets produced. So the spindle fibers, the centrioles, it depends on the type of cell, but all the little pieces of the cell that have to come into place in order for a cell to divide are produced in G2. Okay, and then if you look on up there, you have M and C. Okay, so M is either mitosis, which is a type of nuclear division, where the nucleus and the chromosomes in the nucleus divide, or meiosis, different type of nuclear division. And then C is cytokinesis, the rest of the cell divides, and then by the time you get back up there to 12 o'clock, then you're going to have two daughter cells. Okay, there's one thing that I skipped over up there, and that is G0. All right, a cell's job is not to reproduce. Cells, of course, do reproduce, but they have jobs. Your heart muscle cells have a particular job. Your brain cells have a particular job. Your skin cells have a different job. Your sweat gland cells have a totally different job, right? So cells have jobs. A bacterium out there has a job. A plant xylem cell has a job. All the cells in the world have jobs, right? And their job is not to reproduce. Their job is to do something. That's what that G0 is all about. So at some point in G1, when the cell has grown big enough in size and produced enough molecules, it stops. It stops in G1, and that's what we call G0. So G0 is where the cell is doing its work. All right, now get back to our question about cancer. Cancer cells have this one, well, they have several traits, but one particular trait that is particularly troublesome is that they never stop at any point in the cell cycle, even if something is wrong. Cancer cells just keep dividing and dividing and dividing in that cell cycle over and over and over and over. They never stop in G0. They don't stop in any of the other phases. If there's something wrong, if they haven't grown big enough, if they don't have the DNA replicated correctly, etc. They just keep dividing. And that is what is troublesome about cancer cells. They just continue to divide. Now, there are checkpoints all along that cell cycle that normal cells, if they haven't grown big enough, if the DNA is not replicated, if the cell machinery for cell division is not reproduced, the cell will stop at a checkpoint. A checkpoint's sort of like a gate to check to make sure everything is fine. Cancer cells don't observe those checkpoints, and they just keep on dividing and dividing and dividing. And that's why you can get all kinds of crazy abnormal cells in a tumor, for example. There can be lots of variations. They can be gigantic cells. They can turn into all kinds of strange tissues in tumors. And so that's one of the problems with cancer. Now, also, is it a genetic disease, cancer? Now, have you ever heard of cancer genes? I bet you have. BRCA1, BRCA2. I know you've heard of the breast cancer genes. There are all kinds of cancer genes that give you a predisposition for certain types of cancer. That does not mean that you will develop this cancer, but it means that those genes are there, and if the right conditions happen, then they can be turned on, and you might develop that particular type of cancer. So I'm helping you answer some of your questions here. But I really want you to understand that cell cycle. It's very important to understand how it works and all the conditions that have to be met before a cell can divide. All right, what else do we have? And here's my picture of a turkey. So happy Thanksgiving. That is wild turkey, of course. Happy Thanksgiving this week. I hope you have a great week with your family and friends. I hope that turkey survived when I took his picture. It did when I took his picture. But you may be having turkey this week. So anyway, what else do you have due in week 5? Milestone 3 of the final project. Now this is a different format for sure, because you are starting to make your slides for your PowerPoint presentation. So this week, the parts of the discussion about your topic that you're going to include this week have to be on PowerPoint slides or something similar to PowerPoint slides. Google Slides, I've seen Canva, I can't remember what else. You can just do a PDF with slides. There are all different kinds of ways to do this, but most people have PowerPoint, so PowerPoint slides. All right, this week, what are your methods of conservation? So how can we prevent this problem from happening, okay? If you want to think about it that way. Methods would be methods of conservation. How can we prevent this bad thing from happening? Okay? Education, and why is it so important? Why is it so important to educate the general public about the acidification of the ocean, or all the plastics that are being dumped in the ocean, or air pollution, or loss of biodiversity, etc., etc.? Why is this so important? So you need to explain that. Solutions. How can we fix the problem? Because most likely we already have examples of this problem out there in the world. So how can we fix this? So methods of conservation is prevent, solutions is how to fix. What are the ethical considerations? Please also include laws, regulations, treaties, guidelines, whatever is out there that helps to regulate whatever this issue is. Positive and negative aspects of the issue. Okay, there have to be positive aspects of the issue, even if it's bad. Otherwise, it wouldn't be happening. Okay, there has to be some reason for this to be happening. Say, deforestation of the Amazonian rainforest. It is happening at a great rate. So we don't want it to happen, but why is it happening? Because then farmers have land to grow crops on. You can harvest the wood and sell the lumber. There are all kinds of positive reasons. So you have to think about positive and negative aspects of the issue, and personal impact. How does this impact you? How can you make a personal difference in this particular topic? So that's what you'll be discussing this week. Include citations. And so citations are referencing your resources in your particular body of work. And the references at the end. Use PowerPoint slides or something similar and label all your sections. Methods of conservation at the top. Then tell me about it. Education. Then tell me about another slide. So each of these would be a slide, really. Okay? And then if you want to click off here, because you listen to everything, that's perfectly fine. But I will cover DNA fingerprinting, since this is the week of DNA, and I have always been interested in DNA fingerprinting. In fact, I did my PhD dissertation on this particular technique. I want to talk about it a little bit because it is very interesting, and lots of people are interested in forensic science. So DNA fingerprinting really doesn't have anything to do with fingerprints, and you probably know that. It's also called DNA profiling, DNA typing. And, I don't know what this will morph into in the future, but it probably will have different names in the future. So this all began in 1985 with a fellow whose picture I posted right here, and he has since been knighted, so we call him Sir Alec Jeffreys. He was and is a geneticist in England, and he was working on this particular technique where he was chopping up DNA and then spreading it out according to the size of the pieces that he was getting, and he was looking to see if he could figure out how to tell one person's DNA from another person's DNA, and he actually figured it out. And when he was discussing this with his wife, she said, that sounds like fingerprinting. And so they started calling it DNA fingerprinting. And here's an example of something that he might have looked at way back in 1985. It's called an electrophoretic. It's basically an X-ray of DNA that has been spread out into a barcode. And you recognize the look of that, right? You have seen these barcodes. So he did this technique, and he was looking at families to see if he could see the actual connections between the family members based on their DNA. And he figured out that, yes, he absolutely could. He was publishing his work, and police officials got word of this, and they were having some difficulty in England because there had been 3 murders of young girls in 3 different villages, and they had no suspects. And so the police went to Alec Jeffries and they said, do you think that you could use this new technique to catch whoever this murderer is? And he said, I think so. We can sure give it a try. And so what they did in England was they went to the 3 villages and they collected blood samples from almost all of the men. This was over 5,000 blood samples, and they started sending them to Alec Jeffries. And you have to realize back then it took at least a week to run one of these gels that the DNA is in and take a picture of it like this. So he started running them, and he had the evidence from the crime scenes. They all 3 matched. The evidence samples matched, but he could not find a match for the person who left behind this DNA. And they tested and they tested and they tested. Well, if you're the perpetrator and you hear that they are collecting blood to figure out if you did these crimes, then wouldn't you try not to give your blood? All right, this is what happened. So a fellow went to his friend and said, hey man, I just can't give blood because I've been sick and I just can't. So how about you go, here, you take my ID and go give for me. And Ian Kelly, his friend, said, all right, I'll do that for you. So he did it. Well, Ian Kelly then went to the pub later on and told all of his pub mates what he had done, especially after he had had a few drinks. And one of the women in the group said, this does not smell right. And so she went to the police. And they contacted Colin Pitchfork, who was the fellow who talked Ian Kelly into giving his blood. And that actually is his name. And they tested his blood. And sure enough, he contributed to the DNA at the crime scenes. He was married, he had 2 small children, he had a good job. There was no indication that he would have been the murderer, and now he is in prison for the rest of his life for that. So he was the first person to be convicted based on DNA. But the other interesting thing was that somebody had come forward and confessed in great detail. And Alec Jeffries ran the DNA on that fellow and said, he did not leave the DNA behind. So actually the very first case of DNA fingerprinting exonerated an innocent person, even though he had confessed. But then the perpetrator was actually found and tested, and Colin Pitchfork is now serving life in prison for those 3 murders. So, of course, police departments all around the world heard this, and in 1988, the FBI began using this technique, followed by state agencies, and now we have even county agencies, and so, yeah, there are lots of DNA labs around the world now. The first type of DNA fingerprinting technique is called RFLP. You don't really need to know what that is. Restriction, fragment, length, polymorphism. Anyway, it just means that you chop up the DNA into little bits and they are in different sizes. That was Alec Jeffries' contribution to DNA fingerprinting, and that's an example that you see over there on the right. Next came PCR, and you've heard of PCR, the polymerase chain reaction, because it is the technique that's used, one of the techniques that's used to test for the coronavirus that causes COVID. It's a very quick technique. It uses much, much smaller quantities of DNA, and it doesn't have to be good quality. There's so many benefits to PCR that you didn't have with RFLP. And there are even new technologies being developed now as we discuss this. I think very shortly there will be a handheld unit that you can take into the field and test DNA and run it to CODIS, which is the combined DNA index system that looks for matches. It is coming. And the latest thing that's been happening with DNA fingerprinting is that police departments all around the world have been submitting profiles to genealogy websites to help catch criminals. And the first of these, and most notorious, I guess you would say, the Golden State Killer was caught, Joseph DeAngelo in California. He's in his 70s. He was never caught. I don't think he was ever even suspected. He had been a former policeman, and so he knew what to do to evade capture. And eventually he just got old enough where he couldn't murder anyone anymore. And he has been caught. He is in jail right now, but he hasn't been tried as far as I know. And we have since then used these genealogy websites to catch quite a few more criminals since then. So there's all kinds of things happening with DNA profiling and DNA typing out there in forensic science, but it's also going to be used in places like healthcare. Your physician will have access to your DNA and will be able to look up your susceptibility to different genetic diseases. This is coming in the near future. Also, your ability to tolerate certain drugs, because sometimes people have adverse reactions to drugs that are actually in their genetics. So all of this is coming on the horizon with DNA. So anyway, I hope you enjoyed that. That's just a little bit about DNA fingerprinting. And if you have any questions about it or you want to discuss it with me, please, please, please email me, because of course I love it. All right, so I hope you have a great week. Great Thanksgiving. Let me know if you have any problems. I'll be checking my email the whole week. Okay, m.sigmon@snhu.edu. Have a great week.